Welcome, ladies and gentlemen. So today we are going to start chapter 12, which is going to go over the cell cycle. So here's going to be the thing with this particular set of information. I'm going to give you a lot of additional detail. We're going to go over a lot of the highlights, but I'm not going to sit here and go through what happens in prophase, what happens in metaphase, what happens in anaphase. That is stuff that is typically pretty heavily covered in underclassmen biology, so I'm going to expect that you remember it, or at least are able to go back through the text as you work through things like your reading guides and some of the activities that we do, that's enough to refresh your brain. If that is not the case, then please make sure that you see me. We can sit down individually, but I'm not going to go through all of those details here. We're going to kind of take it a little further and talk about some other things that surround those cell division phases, not necessarily all the details about those phases individually. Okay, so just keep that in mind as we work through this. So, couple of things, right? If we're going to go through cell division, we're going to make new cells. There are a handful of reasons why cell division exists, right? Ultimately, it's going to come down to reproduction. In order to continue life, in order to continue to have the next generation, those cells have to be able to reproduce, and the only way that they can do that is by dividing. When we look at the key roles of cell division in organisms, a lot of it depends on whether or not they are unicellular, so a single-celled organism, or are they multicellular. So across all cells, reproduction is one of the key roles for cell division. When we look at unicellular organisms, that's pretty much their only necessity for making new cells is because they need to go and make the next generation. So they're going to pass on their genetic information and make offspring. When we look at multicellular organisms, we're definitely going to end up seeing reproduction being a role there. When we talk about meiosis in chapter 13, we'll get into some more detail exactly how multicellular organisms use cell division in order to reproduce. But here we're going to focus more on mitosis. And mitosis and that form of cell division really comes down to these three things. I've got a fertilized cell, and now I need to make an organism out of that. So that's sort of the reproductive component here. I've had an egg and a sperm join, and now that single fertilized egg needs to go through rounds of cell division, rounds of mitosis, in order to develop the full offspring. When we talk about an actual adult, or even juvenile for that matter, who is a fully developed organism, then the roles of cell division in that individual become more about growth and repair. So if I need to grow more muscle as I'm getting bigger, more bone as I'm growing, developing, that's one place where cell division is going to become a major component. And then just repair. Cells are going to die, cells are going to get damaged, and so I need to be able to repair them if possible in order to keep functioning. So when we look at those rows of cell division, keep in mind that you aren't just a single cell that continues to get bigger and bigger and bigger and bigger and bigger, right? There's that idea of surface area to volume ratio where the efficiency of the cell breaks down the bigger the volume gets because the surface area doesn't increase at the same rate. And so we go through cell division in order to make more cells in order to maintain that cellular efficiency of things like diffusion and osmosis and getting waste products out of my cells and nutrients in. All of that has to cross that cell membrane, and that cell membrane surface area doesn't increase at the same rate that the volume of that cell will. So eventually we get to a point where the cells get triggered to say, efficiency is dropping here. It's time to make more cells because I can't continue to just grow this single cell anymore. So when we look at cell division, really this idea of dividing cells fits into a bigger picture, that bigger picture being your cell cycle. And so your cell cycle really consists of two major components, and then there's subphases within that, right? So I have my interphase here, and that is the bulk of the cell cycle because it's the bulk of the time that your cells spend in their life cycle. So we've got your G1 here, which is sort of just growth, development, um, doing its normal function. If it's a muscle cell, it's doing its muscle cell thing. If it's a blood cell, it's doing its blood cell thing, whatever that function happens to be. The end of that G1 phase, then you start getting signals, whether they be internal, external, or some combination of both, that it's time to start considering dividing, the efficiency of the cell starting to go down. And so then we get triggered to pass into the rest of interphase, which includes your S phase or your synthesis phase. That's where DNA replication fits in. So we'll talk about that process as well in this unit in chapter 16. And then we'll pass into G2, which is basically final preparations for cell division, duplicating your organelles, making sure that DNA synthesis happened appropriately, that there really truly is a need to go through cell division. And then you end up into your mitotic phase, which gets split into mitosis, which then can get split into prophase, metaphase, anaphase, telophase. And then your cytokinesis is also part of that M phase or mitotic phase where we actually go ahead and split the cytoplasm. So in order for your cells to divide, they have to have a set of instructions, not only for themselves to function, but then for their offspring to function. So when we look at cell division, at least in terms of mitosis, which is what we're focusing on here, that is going to result in genetically identical cells, or what we would talk about as clones, right? So every cell that goes through mitosis is genetically identical, or at least it should be, to the daughter cells that are produced as well as back to the parent cell. So they're all clones of each other. So those cells, when they go through that S phase, they go through that synthesis phase, are going to duplicate their genetic information to ensure that they have a copy of that DNA for both daughter cells that get created at the end of mitosis. And because we're just talking copying cells, that's really the idea here of mitosis, then we're talking about creating that genome and copying it exactly. Now, can things occur throughout that mutations? I should say, can mutations arise as we go through that process? Sure, right? But assuming everything goes well and any sort of mistakes in DNA replication get caught and fixed before the cell goes into cell division, then they should be genetically identical. Now, in order to make this work, when your cells are doing their normal function, so back in that sort of G1 phase of interphase, we are looking at DNA that's all spread out in your nucleus, right? Because we want access to it. But that chromatin form of DNA is not really conducive to cell division and moving things around. So in order to move things effectively, we're going to package that DNA, we're going to condense it into chromosomes. So again, when we look at cells going through just normal function, we see DNA all spread out like this, right? This is your chromatin. And I want to be able to spread it out because I want access to it. It's like you opening up a textbook and being able to flip through the pages and gain information by gaining access to those pages. But if you look, all right, at least in eukaryotic chromosomes, that chromatin isn't just your double helix. It isn't just your string of DNA. Instead, in order to package it effectively in your cells and be able to condense it and relax it when necessary, your DNA is wound around these little balls here, these little histone proteins. So these little purple things here are these histone complexes. So when your DNA is all spread out, this is the idea of chromatin, right? So I can see all those little histones, which is what makes this kind of look fuzzy when we look at it actually under an electron microscope, which is what you're seeing here. But what it's going to allow us to do is allow us to take those histones, allow those proteins to sometimes stick together, right? And that's what this idea of supercoiling is, where I'm basically activating those histones to become sticky. Um, and so they condense and get nicely tight and packed to each other. Or I can allow them to relax and not stick to each other. And really, it's going to come down to, we'll see when we get to gene expression, the histones have chemical groups, methyl groups, acetyl groups, things like that, that will attach to those histones, and that's what's going to dictate their sort of stickiness. The presence or absence of certain groups allows those histones to either stick to each other or not. And so I like to use the analogy of a slinky, right? When I look at that chromatin, it's that stretched out slinky. I'm playing with it, I'm using it, and it takes up more space. But when it's time to move that genetic information, I don't want to be carrying that stretched out slinky down the middle of the hallway between classes, right? It's going to get tangled, people are going to walk into it, it's going to get broken. And so your cells are going to condense that DNA like you would condense that slinky back down to its smallest form so that you can move it effectively without damaging it. Now, you're not accessing the genetic information while it's in that condensed form. You can't. You don't have access to it, just like you shouldn't be playing with the slinky in the middle of the hallway between classes, right? But if all I'm trying to do is move it and move it without breaking it, that condensed form is really effective in doing that. And so your cells will in fact do that. Now, when we look at chromosomes, how many do we have? Well, here's your unduplicated chromosome hanging out in your cells, probably stretched out and being accessed during that G1 phase. We go through DNA replication and we duplicate it. So this is probably what you're used to seeing when we talk about chromosome structure, is this sort of X structure here. And what you're really looking at is a duplicated chromosome. So side note here, that term chromosome, you have to understand the context in which that term is being used. Because we use it in a lot of different places to mean slightly different things. It's all genetic information. It's all the same genetic information. But when we use that term chromosome, we can talk about a single chromosome in a cell being accessed and used. We can talk about this as a chromosome. Truly, it's a duplicated chromosome, but most of the time, most texts, most scientists will just talk about it as a chromosome. And then we can also talk about individual chromatids. And so you've really got to pay attention to the context and when these terms are being used to ensure that you're using them correctly and understanding what they're talking about. Okay, so there's just kind of my side note there. So while we're here, let's talk about some structural things, right? So when we duplicate that chromosome, you do have two sister chromatids, so each side can be considered a sister chromatid. They're sisters in the sense that they should be basically twins, because they should be genetically identical copies of each other. They do get held together here in the center by a centromere, and then that centromere really is kind of a sticky protein complex that keeps these guys together, but then also gets degraded when it's time to separate those chromatids when we go through cell division. All right. And so we'll talk a little bit more about some other structures as we come across them. Something else I wanted to point out here, though, while we were on this slide, is the idea of how many chromosomes exist in a eukaryotic cell. And the truth is, well, it depends on what species you're talking about and what cell you're talking about. When we talk about multicellular organisms, we'll typically split your cells out into two categories: your somatic or your body cells, and then your gametes or your sex cells. And we're going to focus mostly here on these somatic cells here because we're talking about mitosis. We go to gametes, those are going to get created via meiosis, and that's going to be a different process that we talk about in chapter 13. Now, I mentioned it depends on what type of species you talk about in terms of the actual number of chromosomes, right? So we do a quick little designation here, shorthand, so that it's consistent across all species regardless of how many true chromosomes they have. So when we talk about body cells, we're typically talking about 2N cells or diploid cells, meaning that they have two sets of chromosomes. If you remember the term homologous chromosome from your underclassmen course, then we're talking about one set of genetic information you got from your mother, one set of genetic information that you got from your father, right? And they should be homologous to each other in the sense that they carry the same type of information, not necessarily that they're identical, right? So your mom could have given you one sequence for blue eyes, dad gave you a sequence for green eyes, but they are on the same location on the same chromosome in your cells. Your gametes are half that, so an N or haploid. They only have a single set. So that when you go to pass on that genetic information via sexual reproduction, you as the parent are only passing on half of your chromosomes, half the set. Your partner will give the other set to recreate those diploid cells in the next generation. And it's really important that we are capable of doing that so we're not constantly duplicating or doubling your genetic information every time we go through a generation. That would be craziness. Now, if we apply this concept to human numbers, then those diploid cells, your 2N number is 46, right? You have 23 pairs of chromosomes. Your gametes, your haploid number would be 23. All right, so we're going to half that so that you give a single set of chromosomes, 23 chromosomes to your offspring, and then your partner will give the other 23 so that you have a matching set. Now, each chromosome itself contains a single DNA molecule. So when I look at this, this is a single double consistent, continuous double helix. All right, those sister chromatids then get created during that S phase and interphase so that we have a duplicate of that chromosome joined together at that centromere to keep them together until they're ready to split when we go from metaphase to anaphase in mitosis. And each contains the same genetic information. So they are genetically identical to each other so that when we go and split, those daughter cells are genetically identical back to the parent cell, as well as genetically identical to each other. All right, so because we keep kind of bringing this up, these terms clearly are important, right? So let's just make sure we have some actual written definitions for some of this chromosome structure. So I've mentioned the sister chromatids, right? And we can talk about a single chromatid, and then together when they're in that chromosome structure, they are sister chromatids to each other, meaning that they're identical copies of that original chromosome. That centromere here is then that sort of cinched waist that holds them together. It is a protein complex that does get degraded and broken down when it's time for them to separate. And something else I wanted to mention while we were talking about chromosome structure are actually the ends of these chromosomes. And this, I only show you here one, but they exist on every end of these chromatids. And that's this idea of a telomere. Really kind of interesting concept that's going to come back, especially when we get to DNA replication. There's a complication when we go through DNA replication that the DNA can only get replicated so far down the strand, right? So you always end up losing just a couple of base pairs every time we go through cell division, every time that we go through DNA replication. And we'll talk more in chapter 16 about why that ends up happening. But the point here is that those telomeres exist. They're non-coding sort of protective DNA sequences that exist at the end of your chromosomes. Essentially, it's kind of junk DNA. It's DNA that doesn't code for anything, at least as far as we're aware, that is functional in your cells. So that every time you go through a round of cell division, we eat a couple of those base pairs away in that telomere, and we're not eating into good information. Now the question then becomes, how do you get those telomeres back? And the interesting thing is that not every cell has the capacity to replace those telomeres. There's this enzyme called telomerase that exists in things like your germ cells, and those germ cells are what goes through meiosis to create your sex cells. Um, and that enzyme seems to exist there as a necessity because every time we go through and make a new egg or make a new sperm, we don't want that potential offspring that can come from that egg or that sperm to have no telomere, right? So somewhere along the way, those cells evolved this telomerase enzyme to replace it, to recreate it. But most of your body cells don't have that enzyme. Why? We don't necessarily know. Maybe it's a function of energy conservation. Who knows? It just didn't happen. So every time, say, a skin cell goes through a round of cell division, we eat into that telomere. Well, that telomere is only so many base pairs long. So eventually, we start eating into good information. And so there is some thought that perhaps at least some forms of cancer might be a result of the fact that your cells have gone through so many rounds of cell division that they've actually eaten through all of their telomere sequence. They've lost all of that telomere sequence, and now they're starting to eat into good functional information, and as a result have lost the ability to control their cell division, and that's where cancer comes from. So I know at least at one point there was some research looking at, can we force that telomerase enzyme into body cells and potentially fix that telomere issue? And could that potentially be a way to prevent cancer? Who knows? I don't really know where that research has gotten to at this point. Might be something interesting to Google and see if you can find out, but it's just kind of a cool idea at the very least. Because think about it, where do you see a lot of cancers? It's in older people, right? So they've gone through so many rounds of cell division within their lifetime that they have the potential to have eaten through all that telomere. So kind of cool stuff.